In this video, we are going to discuss about antibodies. We know that we have a very effective immune system which is capable of differentiating self molecules from non-self molecules. When our immune system discovers or encounters a non-self molecule, our immune system will produce a kind of proteins which we call it antibodies, which are capable of combining with the antigens and destroying them. So these are the antibodies. We are going to look at the structure of these antibodies. In the initial days, very simple experiments were carried out. For example, if we take serum from a normal animal and carry out an electrophoresis, what will happen is we will get an electrophorogram which will look something like this. So if this is the distance of the migration, and if this is the absorbance, if we take it on the y-axis, what we will get is we will get a kind of a graph like this. So if the electrophorogram will be like this, it will be like this. What each of these represent? So this will be the albumin, this will be the alpha chain, and this will be the beta globulin, and you will find a very small peak here. So this is an example of, this is from a non-sensitized animal. So this is an example of the electrophorogram of a non-sensitized animal. Suppose we inject antigen into an animal and take the serum that is known as the anti-serum. Carry out an electrophorogram or electrophoresis of the anti-serum of the sensitized animal. We will get a graph. We will get an electrophorogram like this. It will be slightly different. So this represents the albumin, the alpha globulin, beta globulin, and then you will find another peak, and that peak is nothing but the gamma globulin. So they were trying to understand what is present in this gamma globulin, and that is how they came across these antibodies which are produced against the antigens. Now, we produce a lot of proteins in our body, but the kind of each protein is unique, and the protein which is synthesized, for example, let us take carboxypeptidase as a protein. Carboxypeptidase produced today, and after 10 years it is going to be the same. There will not be any difference in the carboxypeptidase after 10 years. If the sequences of amino acid will be the same. Whereas when we take the antibodies, we have a capacity to produce huge number of antibodies. Roughly it is estimated that we can produce 10 raised to the power 18 types of antibodies through a process of rearrangement. This is in order to meet the requirements of the varieties of antigens one may encounter in life. So let us look at the structure of these particular antibodies. So if you look at the structure of antibodies, the initial studies were carried out in 1950s. 1950s especially by Rodney Porter. Is a scientist who tried to elucidate the structure of antibodies. So what he did was a very simple experiment. He found, remember, antibodies are Y-shaped structure. We will be looking at the structure. So assume this is an antibody. So in this antibody, what he did was he treated this antibody with an enzyme known as papain. When he treated with the papain cut the antibody, here. So it cuts the antibody here, and what he got is, he got a structure like this. Okay, they are linked by disulfide. This is what he got. And this is the region which is capable of binding to the antigen. This can bind with antigen, and since it can bind with the antigen, he called it the fraction which is capable of binding to antigens. He called it FAB region. The fraction of the antibody which can bind with the antigen. This is the region that is not capable of binding the antigen, but it got crystallized. So he called that the fraction which crystallizes. So these were the fragments which he got. And the FAB region has a molecular weight of somewhere around 45 kilodalton, and this had a molecular weight of about 50 kilodalton. Very, very simple experiment. Another experiment was carried out. That was by using another enzyme called trypsin. Trypsin also cleaves the antibodies. What happens with the trypsin is another interesting story. He got a structure like this. Okay, there are disulfide linkages. Here also there are disulfide linkages plus multiple small fragments. So this fragment had a molecular weight of about 100 kilodalton. So this was in a way trying to elucidate the structure of an antibody by using two enzymes. Another experiment was carried out in 1960 by another scientist known as Edelman Gerald. What he did was, he treated these antibodies with the mercaptoethanol. You know what happens with the mercaptoethanol. Mercaptoethanol will break these disulfide linkages and it will make it a single polypeptide chain. So when a single polypeptide chain is obtained, run it on an SDS page and one will come to know how many polypeptide chains are present. That is how the structure of antibody was elucidated. Basically, a simple experiments carried out in the initial years. Let us look at the structure of this antibody now in detail. So, the antibody consists of 4 polypeptide chains. It has got 2 light chains. Molecular weight of each of these light chain is about 25 kilodalton. There are 2 kinds of or 2 types of light chains possible. One is called a kappa chain, another one is called a lambda chain. In the kappa chain is coded by, in human beings, chromosome number 2. In the lambda chain is coded from chromosome number 22 in the case of human beings. Okay, now there are 2 heavy chains. And the molecular weight of these heavy chains vary roughly about 77 kilodalton. And the heavy chains are coded from chromosome number 14 in the case of human beings. So these are the basic units of an antibody. How do we assemble this antibody? So antibodies are basically glycoproteins. If the shape of antibody is Y-shaped, let us look at the structure of the antibodies. We have seen it is a Y-shaped structure. It has got 2 heavy chains. These are the 2 heavy chains, and there are 2 light chains. This is the basic structure. Now, in the light chains, it has about 214 amino acid. This is the N terminus and this is the C terminus. This is the N terminus and this is the C terminus of the heavy chain also. It has got 214 amino acid and the heavy chain has roughly about 450 amino acids. Now, there is something very interesting about the antibodies. There is a very good analogy given in several books, like you look at the words like immunology, psychology, physiology. You see that there is something very rhyming words here. What is rhyming here is if the word ology is the same in all the three words. Ology is the same, whereas immune, psycho and physio are in the varying portion in each of these words. So these are the variables, varying regions, and these are something called a constant. Antibody, each of these chain also has a constant region as well as a variable region. In this light chain, in the first 108 amino acid, that denote a variable region. In the variable region of the light chain denoted as VL. This portion is the constant region of the light chain, that is a constant region of the light chain represented this way. It is this region and this is the VL region. Similarly, this light chain also has a constant region and a variable region. In the heavy chain also has a constant region and a variable region. This is the variable region of the heavy chain and the remaining is the constant region of the heavy chain. And the constant regions are divided onto, this is the constant region of the heavy chain, that may be CH number 1, and it can be divided into CH2, CH3, CH4, etc. depending on the antibodies. It is divided. And these light and the heavy chains are linked by disulfide linkages. You will find disulfide linkages, and also between the chains you will find disulfide linkages. Within the structure also you will find disulfide linkages. Since there is not enough space, not drawing the accurate structure here, kindly look into a book in order to see how many intra-chain and interchain disulfide bonds are present in an antibody structure. We are just showing the basic understanding of an antibody structure. We have seen this is the light chain, and there are two types of light chain. One is the K, another one is kappa, and the other one is lambda. Both are never found together in any antibody. You can have an antibody exclusively made up of kappa chains or another type of antibody exclusively made up of the lambda chain. 60% of all the antibodies that you find in a system is made up of kappa chain, and the remaining 40% will be made up of the lambda chain. If you look at the constant region of these antibodies, there are 5 different types of constant region. They are mu, delta, epsilon, gamma and alpha. These are the five different constant regions, types. Mu will give rise to an immunoglobulin, which is denoted as IgM, immunoglobulin M. Delta will give rise to IgD, and epsilon will give rise to IgE, gamma will give rise to IgG, and alpha will give rise to IgA. You see, there are 5 different types of antibody: IgM, IgD, IgE, IgG and IgA. These antibodies, all these antibodies are present in our body. What is important for us to know is in what circumstances these antibodies are produced. When we encounter an antigen for the first time, it will be the IgM is produced. When we encounter the same antigen a second time, IgG will be produced.